onward to our second Antonio. Antonio Iavarone is a professor of neurological surgery and
Maybe better,
nice.
right?
He's also the deputy director of the Sylvester Comprehensive Cancer Center at the University of Miami Miller School of Medicine. Before that, he spent 20 years at Columbia. I wonder whether you miss it these days. Last is professor of neurology and pathology and cell biology in the Institute for Cancer Genetics. He was also a research fellow at UCSF before that, Sloan Kettering, and the Albert Einstein College of Medicine. And his MD is from the University of Rome, even though you grew up in Napoli, right?
What is it?
You're from Napoli originally?
Yes, not Napoli, it's another city we don't want to mention. It's very small, called Benevento.
Benevento. Antonio from Benevento.
Thank you. Okay, I hope I did everything right with the connection. Fantastic. Nino, thank you very much for the great talk you gave. That works also as a perfect introduction because obviously I will present to you the work that we do in the lab. But I will start with our frustrated presumption that we should try to have an impact in the clinic. And we think that we have been thinking that since quite a few years, especially because we understand when we study these tumors, as you heard from Forrest, we can understand really at very high detail the biology of these tumors. We understand that the GBM is incredibly heterogeneous at the genetic level, transcriptomic level, proteomic level, really at the multiomic level. Every single tumor looks different from another one, and we can quite accurately these days classify these tumors and come up with some presumptive idea of what could be the best option for that patient. But all this type of thinking has zero impact in the clinical setting. As you heard from Nino, essentially all patients with an IDH wild-type GBM, despite this formidable heterogeneity, they all receive the same standard of care approach. This intervention completely bypasses the potential research efforts that could be used to identify truly precision therapy for these patients. And I will start telling you some low-hanging fruit idea that obviously the precision therapy opportunities in GBM in the clinic of glioma patients have so far failed, and they have really failed also because they have not incorporated some very basic concept, such as whether genetic alteration is a truncal alteration, which is simply present in the vast majority, or as a truncal actually in 100% of the tumor cells, or whether, as for example has been the case for most of the EGFR gain-of-function alterations that have been targeted in the hundreds of failed clinical trials in EGFR. Basically, these are most of the time branched alterations that at best will result in removal of a branch without any satisfactory elimination of the root of the tumor origin for a particular GBM. So the low-hanging fruit for us has been the attempt to identify alterations that represent truncal alterations and that could represent, there are not many in GBM, and that could represent relatively easy target. And one of those that represent actually a great frustration for us, and I'll tell you why, is the FGFR3 TAC3 that has emerged after our initial identification in 3% of IDH wild-type GBM as the single most frequent gene fusion across all human tumors, because at this low rate, this particular fusion has been essentially discovered in many different types of solid tumors. And the reason why I was saying it's being frustrated is the fact that we discovered this fusion in 2012, and as of today, a proper clinical trial in GBM has not been done yet. And the reason why it has not been done yet is because this is present in only 3% of patients with GBM, IDH wild type. And when the standard, I would say, clinical neuro-oncologists hear about 3%, they get terrified. They really don't like this type of trials, and has been extremely complicated to select these patients for a precision therapy opportunity. So these fusions, however, have been studied now, mostly in other tumor types and in basket trials. There has been actually an approval by the FDA of erdafitinib, probably the single most effective FGFR inhibitor for treatment of patients with locally advanced or metastatic urothelial carcinoma. And in the context of these trials, several GBM patients, such as those indicated here, have shown very significant response. So since we arrived to the University of Miami, that was actually one of the reasons why we moved there, was actually to try to inspire the establishment of some clinical studies where FGFR3 TAC3 positive patients, there are also FGFR1 TAC1, but the vast majority of adults with GBM are FGFR3 TAC3, can be pre-selected for the inclusion in treatment with erdafitinib. And you see the scheme of the trial that now is currently ongoing with Macarena de la Fuente, who leads our clinical neuro-oncology clinic. This trial has now recently moved to the dose expansion cohort. We have already enrolled, I think, approximately 12 patients between the initial phase and the expansion cohort. And the results so far have been very encouraging. Obviously, these are interim results, but you can see here some very significant response, actually over partial response, more than a partial response that have been detected in recurrent GBM. The format of the trial here is the standard phase 2, so these are all patients that recur, standard of care. Of course, we believe from preclinical studies, I'm not showing this to you, that the administration of FGFR inhibitors up front might result in even better clinical response. But this trial is proceeding in the context of ETCTN, which is this NCI network, and with them we have to be cautious, so this is the way to start, but hopefully after we complete the trial, we will be able to move to a more advanced schema. Now, the reason why these fusions are exciting genetic alterations is not only because they represent probably the easiest target today identified in GBM patients, but also because they were associated with a very unique biology that allowed us actually to understand some fundamental biology of all IDH wild type GBM. And the unique biology is that these fusions activate mitochondrial metabolism and oxidative phosphorylation as a primary mechanism of transformation, of mechanism of action that actually can also be used in a context of targeting. So, but what we did in the lab after we identified this mechanism of action of FGFR3 TAC3 fusion was to think whether now we could think about classifying GBM based on the biology of these tumors, based on the biological functions, the functional activities that are activated, and as we said, in a heterogeneous manner in these tumors. I'm sure you know that in many other tumor types, the transcriptomic classification of tumors, a typical example is breast cancer, but really there are many other tumors where transcriptomic activities of tumors can help to stratify patients based on clinical outcome, and also, as in the case of breast cancer, also direct patients to completely different type of therapeutic opportunities. This is not the case for GBM. There has been a classical classification that includes these three groups, proneural, mesenchymal, and classical, but really classifying these tumors into these three groups has resulted in very little information in terms of patient survival, inability to recapitulate the tumor heterogeneity, but most importantly, to inability to drive stratification for personalized therapy. So actually with Luciano Garofano, he has been the primary leader of this work. We started an approach that aimed to classify single tumor cells using a single-cell RNA-seq based on pathway-based classification of individual tumor cells. And this was complemented by a bulk classification that was also associated with survival. And this type of analysis led to a new classification of GBM, a pathway-based functional classification of GBM that resulted in the identification of two independent functional axes, one that we call metabolic axis that includes the mitochondrial GBM, exactly the subtype that was initially inspired by the FGFR3 TACC3 fusion, that is countered by a very aggressive subtype that is the glycolytic plurimetabolic subtype, the subtype that is enriched with a high number of myeloid infiltrates, but that is also characterized by the redundant activation of all type of metabolic pathways except the oxidative phosphorylation that it is unique for the mitochondrial subtype. This axis is opposite to what we defined as a neurodevelopmental axis that is actually a differentiation lineage commitment axis that moves from a proliferative progenitor stem-like cell enriched subtype that we call PPR that then is associated with a neuronal subtype that includes tumor cells that acquire synaptic competency for the ability to establish connectivity with the normal neural cells. Now, this classification, as I said, was initially generated from single-cell transcriptome, validated on bulk transcriptome, bulk genetics, but then we had the possibility to have access to the multiomics data from the CPTAC, and you can see that when we aggregate all the different platforms that are available in CPTAC, this work has been done by Simona Migliozzi, who is a partner of Luciano and who recently got a very beautiful baby. And this work, as you can see, basically identifies the 4 subtypes independently for each different platforms with actually complementary but sometimes different features that could not be inferred from the transcriptomic alone. And in particular, I want to point to you, because this will come up again, the identification from proteomics and phosphoproteomics specifically of the DNA damage responses that we found in proteomics and phosphoproteomics, and that we had not identified for transcriptomics as a key hallmark of the PPR subtype. Now, all these type of classifications, of course, can be recapitulated at the single-cell level and at the bulk level, but were initially identified from the whole tumor mass. When we go and try to dissect the different region of the tumor from a multi-regional analysis, from image-guided multi-regional analysis, in which we have the core contrast enhancing and the rim non-contrast enhancing, we could see, here you see from this pseudotime single-cell based analysis, that as we move from the core to the rim, what typically happens as a key hallmark of the transition is the change of the PPR stem-like state of the tumor cells towards a neuronal, highly differentiated tumor cell. And this point has been validated by multiple studies. This is a complementary study. The one I showed you before was a study done from a limited number of cases at the single cell level. This is not at the single cell level, but is at the multi-regional level, done with actually the Mayo Clinic, with Nantran at the Mayo, Arizona. And you can see that a key cluster that we find associated with the non-enhancing rim is a cluster that is massively enriched with the neuronal tumor cells. So this idea that the mechanism of invasion of GBM might be driven primarily towards a transition to a highly differentiated, synaptically competent, neuronal expressing tumor cell state is a concept that is now emerging from many different reports, including, as you will see later, in a proteomic report. But I wanted to show you some data that actually are providing additional evidence for this concept. And these data that we have just recently started to generate are these data that we generate with the CosMx Spatial Molecular Imager platform. That is a platform that uses spatial transcriptomics at the single cell level with a highly accurate segmentation of individual cells, where basically we can clearly assign the transcriptomic feature to individual cells, both tumor cells and non-tumor cells. And as I said, this is a work in progress. Clearly, we have now a number of results that we are trying to solidify, but this is a finding that I wanted to show to you because it's a finding that came from the complete reconstruction of both the bulk, core, front of the tumor and the invasive rim, where you have mostly the normal brain here. And you can see that the only tumor cells that are present actually into the invasive normal brain are actually those classified into the green neuronal tumor cell state. So when we see these cells that are typically scattered, surrounded mostly by non-tumor cells into the normal brain, these cells are assigned to a neuronal state. This is different from what instead happens in the core of the tumor, where you can see that the majority of the tumor cells are classified as proliferative progenitor, and those are the cells that clearly results in the expansion of the tumor mass. So this classification, I alluded before, the motivation for us to go after this classification was to try to understand if we could generate some prediction opportunity for patients following the classification, and obviously then, based on those predictions, also to try to trigger some therapeutic opportunities for these patients. And as you can see here, this is actually a finding that recapitulates what we see with the FGFR3 TAC3 mitochondrial functions of the F33 patients, is that the mitochondrial subtype is associated in the context of exclusively IDH wild-type GBM with a significantly better survival, something that you can see in all the possible comparisons. But more importantly, if we use OXPHOS inhibitors, different type of inhibitors of mitochondrial metabolism and mitochondrial respiration, you can see that we can have significant anti-tumor effects in PDX, in patient-derived xenografts, intracranial PDX, treated with the OXPHOS inhibitors if we use mitochondrial subtype PDX, but this is not the case if we use other type of PDX that cannot care less about mitochondrial inhibition. So Luciano developed a clinical grade probabilistic classifier. This has been published, it is publicly available, and this classifier can be used using typically RNA-seq from both FAP or frozen results. It can be used to retrospectively evaluate. So for example, in the case of Nino, if Nino wants to know if his responses are typically associated in the clinical trial that he described, to a particular subtype, we can use the RNA-seq from the original tumors to determine the association of the therapeutic response with this subtype. But of course, the most important use of this classifier is a prospective use to do patient selection for prospective clinical trial. And I told you that clearly the most mature group in terms of clinical impact is the oxidative phosphorylation subtype, the mitochondrial subtype. However, in the clinical setting, there have been a lot of negative results in terms of testing mitochondrial inhibitors. There have been actually a number of papers that have described the failure of these compounds in the clinic, mostly because of the extensive toxicity that has been associated with these compounds. So we have worked with many of them, but we have identified a compound that is a derivative of metformin. Metformin is obviously a drug that is widely used, a very safe drug. Is not a clean drug. It is a drug that, in addition to inhibiting complex 1 of mitochondrial respiration, has many other functions. But then this compound, lixumistat, has been identified as a derivative much more powerful and more specific of metformin, and has shown a very safe profile in many phase 1 trials that now have been reported. So we basically tested the activity of lixumistat in addition to the activity of the many other OXPHOS inhibitors that we had previously tested. And here you see the patient-derived cells from different tumors that have been classified as either mitochondrial or as glycolytic plurimetabolic. And you can see that while in the glycolytic plurimetabolic, this compound, just like the others, showed very little response, we have encouraging preclinical responses in the case of the lixumistat for mitochondrial GBM. So these are the summary of the preclinical data. This has shown activity against mitochondrial GBM, both in vitro and in vivo, has shown the ability to cross the blood-brain barrier, although not with a concentration in the brain tumors, and is a compound that can be used in clinical trials. And now we have a discussion that hopefully is advanced, is in an advanced stage with Jan Parney and his team at Mayo to do a window of opportunity and possibly also a phase 2 trials to target the mitochondrial GBM in the context of the glioma, the glioblastoma therapeutic network supported by NCI. So this is where we stand with the mitochondrial GBM, but this type of paradigm that was relatively easy. We identified the biological functions, we think about the drug, did not work, simply did not work for the other 3 subtypes. Clearly, the other 3 subtypes are much more complex. So if we, for example, think about inhibiting glycolysis, or we think about inhibiting lipid metabolism for GPM subtype, this was not doing anything, probably because of the redundancy of these pathways in the case of these other subtypes. So with Luciano and Simona, we thought about the possibility of using these clinical proteomic data, primarily proteomic and phosphoproteomic data, to try to deliver at the proteomic level what we had previously done when we were at Columbia, in particular the work we started with Andrea Califano at Columbia a while ago, in which we used the reverse engineering to identify the master regulator transcription factors that are associated with a particular GBM signature. Here what we did is to integrate the proteomic and phosphoproteomic signature, tissue-specific for GBM, and then we came up with the prediction of the kinase network of all the kinase networks that are active are potentially active in the GBM context. And Luciano developed an algorithm that we call Sphinx for substrate phosphosite-based inference for network of kinases. And this algorithm was applied to GBM, of course, but then was also applied to other tumor types in which we were also identifying, I forgot to mention this point, that this classifier, even though was initially developed for GBM, is actually also operational. It can also predict the same subtypes also in other solid tumor types, such as breast cancer and lung squamous cell carcinoma. And you can see that we found some pan-GPM, pan-PPR master kinases, namely kinases that behave as master kinases and primary most active kinase in a particular subtype across multiple tumor types. And these are two of the kinases that really scored at the top as a specific subtype, specific pan-master kinases, PKC delta as a master kinase of the GPM subtype, and DNA-PK, these master kinase for the DNA damage response for the PPR subtype. In terms of the neuronal subtype, the finding of the master kinase of the neuronal subtype was actually inspired from another study that we did with our collaborators in Korea, Jombe Park, because with them we actually collected a large set of matched pairs, primary and recurrent GBM, that we used to do multiomics, including proteomics and phosphoproteomics, something that had not been done in the context of CPTAC. And as you can see here, recapitulating what we see at the spatial level, we see exactly the same type of primary transition, a primary evolution of IDH wild-type GBM when we look on a temporal level. So when we look at the main difference between primary and recurrence matched GBM, what we see is a net expansion of the neuronal subtype of tumor cells compared with instead at the expense of a decrease of the PPR, proliferative progenitor tumor cells. So the neuronal subtype is what we see enriched in the rim, possibly remaining as a resistant tumor cell after standard of care. And this type of tumor state is the one that we recover as the most enriched at recurrence, something that now we have, albeit at a different rate, recapitulated also at the single cell level in the context of a care project with Mario Suva and Itaiti Roche that we have all been doing together. And now, having the opportunity to study the neuronal subtype in recurrent GBM as really a prominent state, we identified BRAF, wild-type BRAF. In this case, there is no mutation of the BRAF gene. It's a post-transcriptional, post-translational activation of BRAF as the top-scoring master kinase for this recurrent neuronal GBM. And as you can see, the use of different type of inhibitors of BRAF results in the recurrent state. In this case, we were using patient-derived cells from matched primary and recurrent states. And you can see that in the recurrent state, we can clearly see the elimination of these neurite extension, these differentiation-related features that are, of course, the hallmarks of the neuronal GBM. But more importantly, what together with Jombe Park we were able to show is that if we now combine temozolomide with the vemurafenib, with a BRAF inhibitor, we could significantly extend the survival of this recurrent neuronal GBM. So this classification can be, as I explained to you, recapitulated at multiple omics level. I didn't talk about the radiomic level. Again, some work that we have done very recently, in which we have identified a number of radiomic features, very specific radiomic features that are associated with these different tumor subtypes. But we hope that, especially if we are able to do a study now in which mitochondrial GBM will be selected in a non-genetic manner. Again, these are non-genetic selections of these tumor subtypes. We hope that at least for the PPR, for the GPM, these type of subtypes can come next as other opportunities for targeting. So I want to finish in the last couple of slides with just telling you about what is the most frequent tumor predisposition conditions in which actually brain tumors are identified, and in particular glioma. Of course, everything we discuss today and in any discussion with glioma is the context of sporadic glioma, but in the case of neurofibromatosis type 1, in which there is a germline mutation of the NF1 gene, we have a syndrome in which there is a very high incidence of brain tumors, in particular optic glioma in children, but also we have a 5-fold increase of non-optic glioma at any age, including the possibility of the development of high-grade glioma in these patients later in life. Now, it's been extremely challenging to study these tumors, and there are very few reports because the surgery is not the choice, the approach of choice in these cases, because of the multiple morbidities that these patients have. So there is a decision to try to have conservative approaches rather than having surgical resection in this tumor. So a few years ago, we created what we called the Landing Consortium that includes now 30 centers across different continents to basically aggregate a sizable number of both high-grade glioma and low-grade glioma from patients with NF1. These are both adults and children, and they can be, in both cases, low-grade and high-grade. And we studied these tumors at the bulk level, both at the genetic level and the transcriptomic level, and then at the single-cell immunostaining level. And we were able to identify two very distinct modules of genetic alterations that characterize the high-grade glioma and the low-grade glioma in the context of NF1. You see the key features here. The high-grade is associated with typically higher mutational burden, as expected, and there is these three group of genetic alterations that include P53, CDKN2A, and ATRX. These tumors are almost always IDH wild type. We actually, I think, found one IDH mutant, but beside that, all of them are IDH wild type, whereas in the low-grade glioma, these type of aggressive genetic alterations are absent, and we instead identify primarily alterations of the MAP kinase pathways that are gain-of-function alteration of MAP kinases. And the other key finding that we reported was the fact that in the context of the low grade, exclusively the low grade, we found a group that we call high immune, because these tumors distinctly present a large number of CD8 T cells, clusters of CD8 T cells that were not otherwise present, that could be distinguished with what we defined as the low immune. So the low-grade group, the low-grade glioma group of NF1 patients can be subclassified into these high immune and low immune. And obviously, this was work at the bulk level. Here you see, again, the characterization of these tumors in which, of course, I think I already told you that the mutational burden is higher typically in high-grade glioma, typically in adults. And here you see the more granular classification of the CD8 T cells in the high immune that are typically associated also with the cytotoxic phenotype that represents the indication that these cells might be able to mount sufficient immune response. Now, again, Luciano, together with Fulvio D'Angelo, who had also worked on the original studies, have recently started to analyze a partially overlapping set. Actually, most of these tumors were completely exhausted, so we had to start again the request for a new collection of cases. But here you see the new cases, 46 tumors that we have been able to analyze at the single cell level, at the single cell transcriptomic level. And here you see the typical classification of tumor versus non-tumor and the microenvironment that is associated in this particular type of NF1 tumors. And this is still work in progress. We are in particular still trying to deconvolute the tumor cell states because we believe that there are some tumor cell states that are unique for the NF1 glioma state. But what I wanted to present to you is really the more accurate, if you want, classification of the microenvironment that now we generate from these tumors, because what we see is that the immunohigh not only has what we had already identified, the presence of these distinct CD8 T cells associated specifically with this tumor, but what we found is that the myeloid cells appear to be characterized different than the typical situation of the high-grade GBM that you also heard from Nino, is instead composed by a pro-inflammatory microglia and macrophages that we believe have a clear cooperation with the CD8 T cells. So these tumors, these LGG immunohigh of NF1 glioma are definitely an optimal subtype for a subtype that might respond to immunotherapies. We also identified an immunoexhausted subtype, again using single cells. We had not identified before, but this subtype has a significant number of CD8 T cells, has a mixed myeloid microenvironment, and is completely different from the immunocold, where essentially no T cells could be identified. The high-grade glioma are very diverse, but they are not substantially different, at least based on what we saw so far from the sporadic high-grade glioma. So we think that for NF1, there is really a specific opportunity for the low-grade, and this is the final slide in which I am showing you the wonderful group of people. There is Luciano here, but he's really representing of a dream team who is in the lab, and many of them moved from Columbia to Miami. I think they are all happy to be in Miami, and all of you who are really tired of Boston, you should write to us, because we have a fantastic opportunity to rescue you in Miami. Thank you.
Thank you, Antonio. Do we have any questions?
Nino.
Antonio, as always, beautiful, beautiful talk. So if I understand correctly, this neuronal subtype is more at the border, and it makes sense that it sets up to recurrences because that's what we cannot deal with surgically or even with radiation, because radiation is pretty focal. You have good drugs against the other subtypes, but those, we have other stuff that we can take care of. What do you think is going to be successful against these neuronal subtypes that sets up to recurrences? Is there something that you can think of?
I think you may know that now there are probably even too many, if you want, approaches trying to target the synaptic connectivity, right, of GBM with the explosion of the concept of brain neuroscience. I think Michelle, I don't know if she's here, but also Frank Winkler in Europe is doing a number of studies. I mean, we have the Perampanel study that is a part of our study here. Of course, the neuronal subtype, we believe, is driving resistance, is ultimately driving resistance to standard of care, right? But it's not necessarily the subtype that is driving the growth of the tumor. The growth of the tumor in the context of the neurodevelopmental axis is coming from the proliferative progenitor subtype, the stem-like cell subtype, right? That's the stem-like cell subtype. So most likely, the association of a neuronal targeting drug with the standard of care that is probably working well against the PPR is really the only subtype that has shown in our hands some activity in the context of the standard care, possibly even with some inhibitors of DNA damage, right? That are those that we think can specifically target the PPR. You see, in that context, probably we can have some positive expectation. I should also highlight that while in the context of this neurodevelopmental axis, we see the neuronal tumor cells infiltrating in the normal brain. By all means, this may not be the only mechanism, right? There could be what have been called the mesenchymal tumor cells, right? Before there is actually extensive literature that the mesenchymal state is what drives the invasion. And this may be true in the context of the metabolic axis. So if we can at least distinguish, right, the GBM tumors into these two very divergent states, right, which are the metabolic and the neurodevelopment, I think we would really make a significant progress. Because this is also a point on which more or less, even with different terminologies, I think the whole world more or less agrees.
And just, if I can ask one more question. How do you put this finding, the different subtypes and their local regional location with Mario's paper showing that hypoxia drives some of these anatomical location in different transcriptomes?
Absolutely. That hypoxia can be the context of the metabolic state, right? Because hypoxia is the hallmark of the GPM subtype. So we have correlated this, right? And as I was saying, we can use a different terminology, but the biology is the same. Please.
I was wondering if you think that the neuronal subtype is driving migration, you think that, signaling is going to be important and, anti-excitatory, anti-glutamate would actually do something. But it also may, impact any immune signaling that may be in the area, right? So there could be a plus and a minus. So have you looked at that? And then secondly, to Nino's point, if you actually debulk the main part of the tumor, wouldn't you expect that you have, by definition, more of the neuronal and the migratory?
Absolutely, absolutely. So on the first point, Luciano, you should really listen to this because we have always found a particular subgroup of microglia which is different from the typical myeloid-derived suppressor cells enriched in the GPM subtype, that instead this microglia subtype, not better defined because, Luciano, this is a great priority, right? Associated with the neuronal subtype, right? So I think that there is absolutely an opportunity there to understand what is the immune state and the crosstalk between these neuronal tumor cells and especially the microglia, which is the primary immune cells associated with the neuronal subtype. In the context of what you just mentioned, I guess the answer is yes, in the sense that if you do a debulking of the tumor, what you remove, what Nino removes, is primarily the core PPR, right? And now you are left with the neuron, right? And in this preclinical model that I showed you, where we actually identified BRAF as the master kinase of the neuron, what we see is that unless these tumors start regrowing, the neuronal state is highly preserved. When they start regrowing, they can go anywhere. At that point, they are completely plastic, right? They can become GPM, they can become PPR, they can become everything. But there, we have lost the battle.
One more question. Forrest mentioned in passing is that there may be an element of collective cell migration. When you look at some of that, is there actually more cell-cell talk between the glioma cells in these, whatever it is, spheroids that show up below the imaging resolution threshold and or with the neuronal, in this case, mostly axon fiber or in the nuclei, there may be some neuronal cells. So is when there is a higher density of these cells in the migratory area, is there more glioma talk or glioma neuronal talk?
I would argue that most of the crosstalk is a glioma neuronal and possibly other non-tumor cells. So let's not forget microglia, oligodendrocytes, possibly astrocytes, right? They could all participate in this crosstalk. In the spatial analysis I was showing you, is that if you look at the peripheral rim, where we see exclusively neuronal tumor cells, right, in the invading front of the brain, the tumor cells are highly scattered, are completely scattered, right? So they are totally surrounded by normal tumor cells. You don't have any neighbor activity between a tumor. If you start having a contact between a tumor and tumor, these cells are starting to grow, of course, right? They are starting to proliferate.